It's easy. So for a completely different talk, we move back to science. So my name's Chris Reid. I've worked in the field of genetic epilepsies for over 20 years. I'm from Australia, in Melbourne. In fact, the first epilepsy gene ever discovered was discovered by a team in Melbourne. So I've had a long history, including a lot of work that's been based around mouse modelling of the disease. So I guess my job here is to provide, hopefully not false hope, but at least hope that we can use these mouse models to help PURA Syndrome. And it really has been a privilege to be in here for the last day or so and to meet so many PURA perfect children. It's lovely to see. So this is the complexity that we're faced with as both scientists and clinicians. PURA sort of broadly fits into the developmental epileptic encephalopathy umbrella, and it very much is an umbrella term. And really quite remarkably and completely unexpectedly, we've discovered that over 1,000 genes can cause this disease. Right? So immediately that tells you that there's 1,000 ways that you can break the brain to end up with epilepsy and development. And what's struggling from a scientific perspective is every way that you break the brain, you break it in a slightly different way. So the best analogy I can think of is a car. There's no point fixing the brakes if you've got no wheels. You need to really understand what the problem is in order to fix the problem. And that's what we're trying to do. And I guess my hope is that we can use animal models to try and at least achieve that to some level. And obviously today's focus is PURA. So how do we make a mouse model? And this is an important slide. How many here have heard of CRISPR technology? Is it something? Yeah, so it's become very, very common. Now clearly CRISPR technology has been used in numerous ways, including therapeutics, but the way that we've used it in our laboratory is to develop mice models of the monogenic epilepsies. And essentially it's a molecular scissors. What we do is we find the PURA gene, chop it, well not we, it's the royal we, somebody else does this, chop it out and we put in the same mutation or variant that's found in a patient, or at least one patient. And I think what that gives us is a sense that if this mouse, if this mouse here behaves like we would expect it to behave, we can be reasonably confident that mouse is doing something similar to what's happening in the children or adults that have the disease. And by the way, if anybody wants to ask a question, please put your hand up. My hearing's not great, so you might need to talk loudly, but if something I've said that doesn't make sense, feel happy to engage. All right, so we have a PURA mouse. There are numbers, I think people have heard that over the years, but this is one that we've specifically made. To the best of our knowledge, it's the first knock-in mouse model that's based on a human variant. That is actually a picture of the PURA mouse. And from a scientific perspective, the first thing that we want to know if we're going to study this for epilepsy is if the mouse has seizures. And thankfully, from our perspective, it does. So this PURA mouse, if you look at this thing here, what you can see is an EEG, so we're recording from the mouse brain, and you can see this massive tonic-clonic based seizures. So these mice have seizures, but they're relatively rare. So a little bit like your children, they often have them in clusters. They'll have them not for 3 or 4 days. Sometimes some of the mice don't have them at all. And that's actually a challenge for us because it means that we can't easily test drugs, exactly like it is in a human context. So one of the things that my laboratory's done, and I think we're unique in this regard, is focus on another measure of excitability inside the brain. And that's these epileptiform activity. So for most patients that get diagnosed with a DEE, this is across the board, epileptiform activity is a defining diagnostic marker. So it's a real, there's a correlate to that in the human context. The nice thing about these is that we can get lots and lots of them in a given period of time. So we can get 20 or 30 in a 30-minute time frame. What that means, we can count for 30 minutes, we can give a drug, and then we can count for 30 minutes afterwards. And that's tractable. I think everybody can see that it's tractable. And what I'm going to show you next is how we can use this marker here, to potentially give a prediction for drugs. So this is the question that we're asking. Can we use mouse models to predict what anti-seizure medication is best? And I have to say here, we haven't done that for PURA yet, right? I think Mel just told me 2 minutes ago that we had the PURA mouse for 2 years. It takes a long time to breed them, to get ethics, etc. So we're in a position to do this currently. So the example I'm going to give you is another mouse model that we've made, which actually has shown very, very categorically that we can use these mouse models to predict anti-seizure medication efficacy. So this is little Ebony's seizure chart. This is her dad. I'm sure a lot of you have done this previously. And Ebony was placed on a drug called lamotrigine. Most of you would have heard lamotrigine somewhere in the field, those certainly that have been involved with epilepsy. It's a very commonly used anti-epileptic drug. And in fact, it makes sense because the epilepsy we're looking at here is somewhat focal, and that's the first drug that clinicians will use. But what Tim noticed was that as the dose increased, we got progressively more seizures, right? And at this point, we don't know, well, Ebony and her parents and clinicians didn't know whether this was going to happen anyhow, whether the clinician, whether she had a fever, which would have made things worse. But what we did is when we injected that into our mouse model, we see these massive increases in the spikes I've talked about. And in fact, in this case, lamotrigine actually caused seizures. So we can say that lamotrigine, which is weird, because in most of this audience here, lamotrigine would work, actually causes seizures. And Ebony was taken off the drug and obviously improved. Now, there's only one Ebony, but we can make as many of the mice that have Ebony's variant in. So we can make as many as we need. And what that means is we can now systematically test anti-epileptic drugs. And most of you that have been involved or your children have had seizures would note that most of these are drugs that you get off the pharmacy shelf as we speak. And what was remarkable when we did this is we noted that some drugs work and some actually made things worse, okay? And this is incredibly difficult to do in a clinical context. Different children, different ages, very difficult to know whether things are working. And for most people, it's polypharmacy. This drug doesn't work, so you add another drug, etc. But in a mouse model setting, we can do it much more cleanly. The question then is, well, is this predictive? And we showed that for Ebony. But this is a lovely story and one of my highlights as a scientific career. Tim's here, and this is Fern, has the same mutation as Ebony. So the mouse is relevant to her as well. And we had a conversation because people had heard that we were doing this research. And the last bit of that conversation was, what drugs are you on? And she was on lamotrigine as well. And she came off lamotrigine and went from 10 seizures a month, which is significant, and she has effectively been seizure-free since. Ebony still has seizures, so this is not perfect, right? But it tells you that, at least in these children, we shouldn't be seen, shouldn't certainly be given lamotrigine. It's not just in Australia that we had the impact. This is a website. You're all too familiar with the desperation that some of these diagnoses arise. And this is a response from a parent in South Africa. And that was in response to a paper that we did. So we know in this case it was carbamazepine, another drug, but another sodium channel blocker that was making things worse. So the nice thing about this is that we can confidently say, at least in this mouse model, we don't know for PURA, but in this mouse model, that it's predictive. So there's two things that's good about that. It's told us what off-the-shelf medicines to give, but also with new medicines coming on, we can say if it works in this mouse, it probably will work in Ebony and Fern. All right, so the next part is something that's going to take us a longer, and Pew presented some of this work yesterday, and I'm not going to go into huge detail around this, but, oops, sorry, go back. What we want to, that's all right, we can go from here. What we want to do with this mouse is obviously understand what's causing epilepsy inside the brain. Now, your brain is an electrical organ. It's working on a microsecond to microsecond basis, so it's electrical in its nature. So we have to record electric activity in the brain, and that's what Pew does. So what you can see here is literally a needle that's as thin as a hair, even thinner maybe, with a small tiny hole in it. And that tiny hole gets put on a cell, which you can see, if you probably can't, but that one's a cell there. And that gets put on that cell, and then Pew literally sucks a little hole in that cell, and you have electrical access. And what that allows us to do is measure the electrical activity of that individual cell. And we know from other models that these electrical activities are different. and that will give us clues as to what's causing the epilepsy in PURA syndrome. And this is work that Pu's doing literally as we speak. Well, not now because she's in Munich, but when she's in Australia. So people understand that. So that this is a perception. I mean, this is important because what it hopefully gives us is clues as to what to use potentially in a repurposing. If we know what's different between the two, we can potentially, and I do say potentially, work out drugs that might be useful in that context. So obviously we've talked a lot about what different types of therapeutics that we're going to try. We have a biomarker for seizures, right? That's exciting. We already have something that we can measure, and we can put something in the mouse and test it before it goes into children with PURA syndrome. Not work that we'll be doing directly, but Dirk is talking, and others have been doing this already in terms of genetic therapies. We won't be making the genetic therapies, but we can test them. Hopefully through repurposing, we can get, through screening and trying to understand what potential drugs, and that was beautifully described in the talk earlier, and obviously the disease mechanism targeted. So these are all approaches that we'll take in real time, and hopefully, for PURA syndrome, we get a hit. So the next type, so that's going to be our primary focus, obviously, is epilepsy. But what I want to get across, and you guys all know this already, wherever PURA is expressed, you would expect that organ system to potentially be impacted, right? It's logical, and we sort of know it, and you know that from your children. And we know that PURA is in the heart. We know that it's in the cerebellum, in a different part of the brain. We know that it's in the eye, and we know that it's in thalamocortical networks that are important for sleep. And what I want to try and convince you here now is that maybe these mice models, with the same concept, we can only make, there's only one ebony, but you can make as many ebony mice as we need. We can start to test and look at these things and actually maybe give clues as to what might be going wrong in these different systems, with the view that we can not just treat the seizures themselves, but treat a lot of the other aspects of what's going on in PURA syndrome. So this is the first thing, and this really relates to cardiac aspects. So one of the things that we've already discovered, and it's a very robust finding, is PURA hearts are slightly bigger. And there is some evidence that there is some congenital changes. Perhaps more importantly, what we do know is these mice die of seizure-related deaths, and I know SUDEP is a massive issue in the community, and rightly so. We think that these premature deaths, like seizure-related deaths, are a model of sudden unexpected death in epilepsy. And work done not in the PURA model, but what work again that we will be doing moving forward, is to use this premature death as a way of measuring a SUDEP susceptibility. And in really quite exciting data that was done in my laboratory by Ming So and others. This is not PURA, just highlighting that. But the same sort of scenario, these mice died prematurely from a seizure-related event, and we know that they have a heart defect as well. What we did is we used a drug called atenolol. I don't know if anybody here knows atenolol. It's a beta blocker, so probably about 1 billion people on the planet have used atenolol, so it's an incredibly safe drug. It's used to slow down the heart. And our logic here was to protect the heart of these mice in the hope that it would protect the mice from dying prematurely. And when we do that, you can see that there's quite a remarkable reduction. So this may or may not be relevant to PURA. We need to test that, and that's where mice come into it. But what we hope we have here is a way of repurposing the drug that's available. We know this drug can go into paediatric patients, and we're already starting to seriously talk with clinicians about doing at least some preliminary data on this. But the idea here is that protecting the heart from a seizure actually can reduce mortality, and it's something that we are moving forward. Hugh showed this yesterday, but for those that didn't see it, we also know that PURA syndrome can't work as well, and I think we've heard a lot of that already. That's a key issue with PURA syndrome. I always like talking through some of these methods. People looking a little bit weird, but if you get a mouse to walk up a ledge beam, if they're not very good at coordinating, they'll start to slip a little bit. And if you just measure those slips, you can get a measure of how good they are at walking. And you can see here they have many more slips. This is a rotor rod where they get on a rotor and they go really, really quickly. The mice fall off when they can no longer keep up. And you can again see, this is actually truly, this is PURA mice here. You can see that there's a reduction so that they fall off quicker. And this is actually much more complicated. You actually look at the gait itself and you can see there's a deficit. So these mice not only recapitulate the seizures and the SUDEP-like factors, there's also some of the other things that are affecting your children, including gait analysis and stuff along those lines. This is also very exciting work. I know a lot of PURA syndrome patients do think may have visual issues, and that was nicely demonstrated before. Again, we know that PURA is in the eye. This is a complicated diagram, but these circles essentially mean that PURA is expressed in all these different cell types in the eye itself. So it's not going to be surprising that the eye might be different. What's lovely about this is that the eye is a bit like a brain on a stalk. That's how it's been described before. It's got all the complex networks, but it's very easily accessible. You can put a flash of light like this, and you can get a very characteristic signature. And what we've already shown in clinical studies and in our mice in Dravet syndrome, which most of you would be aware of, that there is quite a significant deficit. And what's potentially nice about this is that we can use it to measure drug responsiveness as well. And this is something that we're thinking that just keep your ears out for this, or your eyes, because it's something that we're going to try and do with, in collaboration with an optometrist in Australia. And then finally, this is the last slide. We can measure sleep disturbances. In this case, this is the mouse that has the ebony variant in it. And in this case, she doesn't go to sleep at night. It's really, really quite distressing for the parents. I don't know, PURA is more like sleeping more than less. But again, what you can see here, this is just how long they're awake. And you can see that the mice, you've got to remember that mice go to sleep when they go into the light, as opposed to going into the dark. So this is dark, and as they move it into the light, normal mice get sleepier and sleepier and sleepier. The mutant mice don't. So this is, again, a recapitulation of what we're seeing in the humans. And we can test drugs and have tested drugs on this as well. So that's it. I think we really are wonderfully positioned. We have a mouse that has phenotypes that are important to you guys. We have the capacity to breed as many as we need. Obviously, we have the capacity to do so many different types of experiments, and some of those have been highlighted before. One of the first things we are, and that's happening now, is to look at standard of care, just so that potentially provide a framework. We want to understand, we're scientists as well. There's something really intrinsically nice about understanding why PURA causes seizures. But from that, I know we can also help develop better therapies. We have the capacity to look at cardiac, eye, sleep, and also gait. So all of these things are potentially possible in a mouse model. And really that just leaves me to thank a lot of people, including a huge call out to the PURA syndrome. They've, as you all have heard, they're going to be giving us some money to do the electrophysiology studies and some of the other bits and pieces as well. PURA Foundation Australia, which helped, well, didn't help, actually made the mice in collaboration with Phenomics Australia. And obviously, that's ongoing. This is a fantastic collaboration with PURA Foundation and the Flora, which Mel nicely showed. And of course, I'm the front man. The people that actually do the work are mostly in here. And a big call out to Jenna and Pew and Ming in particular. So thank you very much.